We're going to review transcription before we get started in translation. So what's happening, again, in transcription is we're making a copy of a section of our DNA, and that copy is going to be made out of RNA. So we're going to have a template strand of DNA that we're copying from, and the enzyme that we're going to use to make this process happen is RNA polymerase. So RNA polymerase has kind of a similar role to DNA polymerase in that it's going to attach to the DNA, to a template strand, and it's going to direct the binding of different nucleotides. So DNA polymerase directs the binding of a DNA nucleotide to the DNA template strand, and it just is going to stay bound. What RNA polymerase does is it directs the binding of an RNA nucleotide, which we know is different, basically they have a different base, and it has a different sugar. So it's going to direct the binding of the RNA nucleotide to the template strand. But then after it binds, it's going to feel free to just kind of fall right off and like wave around inside the nucleus. It's not going to stay bound because the bond between the RNA and the DNA is not as strong as that between the DNA and the DNA. And this whole process is going to happen at a region of the DNA called the promoter. That's where RNA polymerase is going to first attach and begin transcribing the DNA. So the attachment of the RNA polymerase to the promoter is initiation. And then in elongation, we have the creation of this RNA transcript, this mRNA transcript, that's going to hang off of the DNA. And this is really what we wanted to create in transcription, is this transcript. The goal was to make this RNA copy. And so the RNA polymerase is going to just keep traveling down the DNA until it hits what's called the stop sequence. So that's a section of the DNA where the polymerase is not going to bind as well, and it's just going to fall off the DNA. So it's going to fall off, and we're not going to be transcribing anymore with that polymerase. And so after it falls off, now we have what's called an mRNA transcript. So is that mRNA transcript ready to just leave the nucleus and go make some proteins? Or are there more steps that have to happen? Any guesses? What's that? Yeah, there's more steps. Yay. So we begin with the mRNA transcript. This just came off the DNA, like fresh off the press, it's brand new. And now, so we're going to have to modify that. How are we going to modify it? Any guesses? There's some hints on the slides. Anyone want to choose one and explain it to me? Because we've kind of been through this a little bit before.
Yeah.
You can do it. Focus. What are some of the steps that are happening in the processing? Yeah, stuff's being cut out. So are we just kind of... Exactly, yeah. So we're going to cut different things out at different moments, different types of transcription. So just because we have the same mRNA transcript that we copied from the DNA, from the region of the DNA between the promoter and the stop sequence, we have this transcript, but we can cut different parts out of it at different times. So we have regions called the exons and regions called the introns. So introns are what we cut out. These are the introns. They're kind of floating away. They're no longer on the transcript, on the mRNA. And so these are called in the way, or introns. Introns are in the way. They're not going to be coding for proteins. So again, the whole point of this process was to make more proteins, but the regions of the mRNA that we cut out, called the introns, aren't going to be used to direct protein synthesis. And so we can use a process called alternative mRNA splicing, or I mean, the cells use the process. And alternative mRNA splicing means that we can cut out different regions at different times to have a different final product of mRNA, even though we started with the same transcript. So this allows us to get more information out of our DNA. So just because we copied the same region of the DNA in between the promoter and the stop sequence, it doesn't mean we always have to end up with the same final product, the same final mRNA version, because we can cut out different regions at different times. So we have another slide on alternative mRNA splicing, but before we go on, what are some other types of processing that happen? We have a hat of types. What is going on here? Yeah, cap. Where's that going? And what does it do? Come on, participate. I would just get goofier if you don't participate, and that won't be good for anyone.
He's the leader.
It's the leader, yeah. So our 5' cap goes on the front of our mRNA, on the 5' end of our mRNA, and that's going to tell other biomolecules and organelles where the beginning of the mRNA is. So it helps us kind of orient the mRNA. And what's the significance of this this tail at the end, the poly A tail on the 3' end? Why do we have that? Yeah, so what does this mean? Why premature degradation? So it's going to eventually degrade, but it's just going to take a little bit longer. Why would we do that? Yeah, so it has time to get to where it needs to go, and then we'll always have to make new mRNA. So if we didn't have, this ability to degrade our mRNA, that would mean after we make it and put it in the cytoplasm, it's just always going to hang out there and always be used to create more proteins. But instead, it degrades. But the longer this poly A tail is, the longer it's going to take for our mRNA to degrade. So if we want this mRNA to stick around for a long time and be used to make more and more protein, we're going to make a longer poly A tail. If we just want to make protein in a very short period of time, we'll have a short tail and it'll get degraded quickly. So this is a way that we can control how much protein we make. So with alternative mRNA splicing, we can change the types of proteins that we make. And with this poly A tail, we can control how long we're making it for and like how much of it that we make. Because we know that the longer that it's there, the more protein that we'll make, and that'll either catalyze more reactions or just change the chemical environment of the cell in a very specific way, more or less, depending how long it's there.
So the introns are in the way. What are exons?
Oh, great. So exons are the regions of the mRNA that end up in the final product that get what we call expressed. So we say DNA is expressed when we make the protein that it codes for. So DNA, again, has this information for making different proteins, and we'll say that gene or that region of the DNA that makes a protein gets expressed when it's making more protein. So the longer the poly A tail, the more protein that we make, that gene is getting expressed more and more. These are ways that we can control the expression.
So the bottom poly A tail
Yeah.
is getting less expressed because it's smaller?
No, so this is just, this doesn't have like information for making proteins. This is just a type of base that's added to the end that gets chewed up over time. So the longer it is, the longer it takes for the cell to chew and break it down.
Like a bunker.
What's that?
Like a bunker?
Yeah, sort of. Like it kind of resists changes to the mRNA. Yeah. But you can kind of think of it as like there's like a Pac-Man that just like chews on the end of the mRNA. And the longer the tail is, the longer it takes that Pac-Man to get into the important regions of the mRNA. The shorter it is, the sooner it gets eaten up.
But like, how do we control like
Great. Good questions. So kind of over the semester, whenever we bring up proteins, we see that cells are always behaving differently in different environments and different times, but they all have the same DNA. So it's like how do they behave differently if they have all the same instructions? And so what we're going to learn about next chapter is how we can use the same common instruction manual for all the cells to do different tasks at different times. So like a long story short is when you're making the protein, there's some signaling that goes on from the external environment to tell the RNA polymerase how long to make this poly A tail. Because the cell knows like, okay, I have this environment, I want this protein to be there for a really long time. Let's give the mRNA a very long poly A tail. If it just wants to make a short change, then it says, okay, we just want to give it a short poly A tail, and that way it'll make a short change.
So a poly A tail basically
it controls how long it takes the mRNA to get degraded.
Okay.
Yeah. All right, so on to alternative mRNA splicing. Again, we can start with the same instruction manual, the same DNA, and we can make different proteins at different times if we can control this alternative mRNA splicing. So now the cell has the ability to read the same instruction manual and then splice the mRNA differently, cut out the different introns and leave different exons to make a different protein. And it can do this according to its chemical environment. So this is how we can kind of regulate the transcription and translation process, one of the ways, and we'll talk more about it next chapter. So also our guest lecturer is going to talk a little more about introns. So in the book, and to the best of my knowledge, introns do like nothing really useful for the cell. But it turns out if you study them a little more closely, you can see that they actually do have essential functions that are not really clear with this perspective, but our guest lecturer is going to talk about why introns are there and how they're not just kind of like a waste of space within the DNA. All right. So what's the point of transcription? Why are we doing this? Great, to make an RNA molecule from a DNA template, and that RNA molecule can leave the nucleus, and the DNA molecule doesn't. So where does transcription occur then? In the nucleus, exactly. Where does replication occur? Also in the nucleus. Where does translation occur? No, it occurs in the cytoplasm, out of the nucleus, because our RNA leaves the nucleus. What enzyme is involved in transcription that we've talked about? What? Yeah, RNA polymerase. So we have RNA polymerase in transcription, but we have DNA polymerase in replication. So they're different enzymes. They perform similar roles, but also non-interchangeable roles. They're different roles as well. So how, speaking of the similarities and differences between RNA and DNA polymerase, how is transcription similar to replication? What are we starting with in both replication and transcription? So we're starting with a template strand of DNA. So in both transcription and translation, we're starting off with like one strand of DNA that we're going to make a copy of. Which
101 to make a new 101.
Yeah, exactly. Great. So it's also semi-conservative. But in replication, we make a DNA copy of that DNA, and that just ends up being a double-stranded section of DNA. It's just we have two identical copies. But in transcription, we don't make a DNA copy of the DNA. We make an RNA copy. So RNA polymerase is different from DNA polymerase in that it makes an RNA copy of the template strand and not a DNA copy. And again, so why don't we send DNA, this should say out of the nucleus.
Because we don't want it to be damaged.
Exactly, we don't get it damaged. It's very fragile and important to us.
Did it even get out?
Yeah, I mean, it kind of does during mitosis when we break down the nuclear envelope. So it kind of like floats around out there, but the cell is not super happy about that. It tries to make it quick. We can have like viral DNA in our cells, so another type of DNA in our cells. So we can have DNA in our cytoplasm, but it's generally not a sign that things are working well or according to plan. All right, now let's get started on translation. I know you're all super excited. It's your favorite topic. What's that? Starving from jumping?
No.
This would be a unique situation to be in.
From jumping up and down.
Oh, that makes more sense. Okay, so replication, we're copying our DNA. Transcription, we're making a copy that leaves the nucleus. And now translation, we're taking this RNA and we're making a protein. And finally, proteins, we know are biomolecules that really, they do something. They change their chemical environment. We know when we talked about enzymes that if an enzyme is present, it can really accelerate or catalyze a chemical reaction. And that's going to allow a cell to change its chemical environment to make it kind of more conducive to that cell surviving and growing and reproducing. So proteins are how the DNA really changes the cell's chemical environment. So the DNA just contains the information, but the proteins are really like kind of doing the work. And so we're kind of translating from one type of biomolecule in RNA to another type of biomolecule. So that's maybe the way we can think about translation. So transcription, we're just making a copy, so it's from DNA to RNA. But translation, we're making a totally new biomolecule. And again, as we control the sequence of our proteins, our primary structure, that primary structure is going to control how the protein folds and its tertiary structure. And that tertiary structure is going to dictate its function, like the way it interacts with other biomolecules, other molecules in the chemical environment of the cell. So by controlling the primary structure of the protein that we synthesize, we can dictate its tertiary structure and dictate its function. And so this is really how the cell is able to change what's going on. Does that make sense to everyone? All right, so we have our mRNA that left the nucleus, and onto that is going to attach a tRNA. So this is another type of RNA. So again, these have ribose as a sugar. They have U rather than a T base. These are single-stranded, not double-stranded. And this tRNA is going to have what's called an anticodon that binds to a codon in the mRNA. Has anyone heard the terms codon and anticodon before?
Yeah.
Great. What do those mean? What's that? I'm sure there's some medical devices that.
It's how you call it?
Oh, cool. Okay. Yeah, it's a different type of codon. No, but I'm sure they have a similar root. I'm just not sure.
You gave everybody here a codon, you know it.
I will take your word on that. I think I would just get more questions than answers. Okay, so on our mRNA, we have what are called codons. And so these are sets of 3 bases that we're going to use to match to an amino acid. So let's just kind of illustrate why we need 3 bases. So how many types of RNA nucleotides are there?
4.
It's a weird day. Okay, so there's 4. And what are they again? I forgot. You did? What? A, G, C, and U. There's 4 types. Did I get it right? I think I did. Oh, wow, that sounds good right now. Okay, but how many, do we know, is there anyone off the top of their head how many types of amino acids there are?
Like a lot.
Yeah, there is. 20 types of amino acids.
I was close. I see it right.
So if the way we kind of coded, the way we translated from mRNA to amino acids just included looking at individual bases, we could only really ever specify 4 types of amino acids. We could say, all right, if you're a G, we're going to bring over this amino acid. If you're an A, you get this one, U, that one. And that really wouldn't be enough to express all 20 types of amino acids that we have in the cell. And so all these different types of amino acids are necessary because they all behave differently. And so they can all give a different tertiary structure. They give the protein a little more flexibility in taking up different shapes because they can bind to each other differently. They can bind to other molecules differently. They can really do a lot of unique things. So we really need all 20 amino acids. So we can't use just one mRNA because that would only allow us to specify 4 amino acids, and that wouldn't be enough. What if we use two bases. So what if we just check and say, okay, if you're CU, you get this amino acid. If you're CC, you get that amino acid, and so on. How many amino acids could we encode for then? So it would be, gotta put any one of the 4 in the first position, and then any one of the 4 in the second position, and that would give you 16 possibilities. So with just looking at 2 bases, with kind of like a little reading window of just 2 bases, you could encode for 16 types of amino acids, but this still isn't enough. We would still have 4 that we couldn't express, and that would really be like a big loss to protein function. It would really decrease the amount of different roles that a protein can play. So if we include 3 bases that we look at, now it's 4 times 4 times 4. And that's 64. So now we can express all the possible amino acids, and we also have some kind of like duplicates. So some types of codons, or sets of 3 mRNA bases, will be duplicates. So they'll express for the same amino acid. But we need 3 to be able to express all 20 amino acids. So we look at a set of 3 bases on our mRNA, and that's called the codon. And what attaches to that is the tRNA. And so on the tRNA, we have what's called an anticodon. And so the anticodon comes in and it binds to the codon, and as it does that, it's going to bring over an amino acid. So we have one type of tRNA molecule for every amino acid. So we have 20 types of tRNA, and they're going to have different anticodons that can come and bind to our codon. And so that's going to allow us to translate the information from our mRNA into amino acids. Because we have different tRNAs that carry the different amino acids, and the tRNAs can bind to the mRNA because they undergo complementary base pairing, which we talked about last week. Complementary base pairing is the set of rules that dictate which bases can bind to each other. So are there any questions about translation?
So with the duplication of the amino acids, you mentioned that those duplicated.
Yeah.
Is that where is the duplication what corresponds to the anticodon versus the codon?
Here, that's on the next slide. I think this figure will help. So what we have here is these different colors represent different bases in the codon. So we have 3 different sets of bases: first base, second base, and third base. So here is like if our codon was GUU, we'd have G as our first base, U as our second base, and then U as our third base would be GUU, and that's going to bring over a valine. But GUC would also bring over a valine, and so would GUA, and so would GUG. So to get a valine, we can have any of these codons. As long as it starts with GU, it doesn't really matter what the third base is, we'll always get a valine. But that's not true for all of our codons. Like here we have GAU and GAC codons bring over an aspartate, but GAA and GAG bring over a glutamate. So here the third base does matter a little bit more. But this is the code that kind of like the way that the cell translates from mRNA codons, so sets of 3 bases in the mRNA, to amino acids. And then this allows us to control our sequence of amino acids, our primary structure, from our mRNA, which again we copied from our DNA.
How do you know where to stop the translation?
Great, excellent question. So when it gets to the, translation or, yeah. So when it gets to the stop codon is when it's all going to stop translating. We'll talk about that on the next slide. It's a good question. But when the, so there's a video that'll illustrate this a little bit better than I'm going to. But what happens is, we have our ribosome that's going to be reading our mRNA. And when it gets to a stop codon in our mRNA, which is they stop those 3 codons that start with U, A, or UC, UG, sorry. My vision is so bad. When it hits the stop codon, it's all going to break off and going to stop translating that mRNA.
So would you give us a chart IDs and make us read?
Yeah, so we're going to do that a little bit on the review. I'm going to give you sequences of mRNA and ask you what amino acids they correlate with.
That stop codon, is that also like a cap?
So what do you mean by that?
The cap, earlier when we were going over the mRNA and transcription, the cap was like the beginning of.
Right. Not so much. So I see your question. The cap was more about like preventing premature degradation, but the stop codon is kind of like the opposite. It's about stopping like excessive translation. But it just makes everything stop happening. It makes like the ribosome break away from the mRNA, and we stop elongating our protein. So I just wanted to talk a little bit about the steps involved in translation, and then do an overview, and then we can watch like a summary video. Sound fun to everyone? The 3 steps of translation, we have initiation, elongation, and termination. Very similar to what we had in transcription, but they're a little bit different here. So at initiation, yeah, but just think about what the process is overall. So a translation, you want to end your translation. We're starting with an mRNA, and that mRNA is going to get attached to by a ribosome, and the ribosome attaches at our start codon. So we saw on the chart before there is one codon that's called a start codon, and that's where the ribosome is going to attach. And then it's going to basically slide down our mRNA, and it's going to add more and more amino acids each time it gets to a new codon. So it'll read 3 codons and move on. And so the way that's going to happen is our tRNA is going to come in. It's going to be carrying its little amino acid here. It's going to bind to our mRNA. So it's going to match its anticodon to the codon of the mRNA. And then it's going to attach its amino acid to this growing what we call a polypeptide chain, so like just a string of amino acids. So we went from this mRNA, and we used our ribosome to add amino acids to this chain, and then at the very end we have like a long chain of amino acids that's going to fold and make a new protein. And we can have multiple ribosomes attached to the same mRNA at one time. So we can have this one little bit of mRNA, and we can have many ribosomes attached to it. That's sort of what we see here, because you have, this is like an actual photograph of one mRNA with multiple ribosomes attached to it, just translating that mRNA. And it's all going to stop when the ribosome hits the stop codon, and then everything's just going to fall off. And then we have our protein that just floats away and goes and does its thing.
How does the correct tRNA like go to the enzyme? How does it know which is going to need?
So what's happening is tons of incorrect tRNA are coming in and bumping into the mRNA, but they're not going to bind to that codon. Their anticodon won't match, and so then they'll just leave, and nothing will happen.
So it's not like it just shows up and then leaves. It just
Exactly. It's just like random collisions. Everything in biology is just like a bunch of random collisions that are somehow orchestrated perfectly to make really complex systems. But that's a great question. I mean, like, how does it know I need this amino acid? It doesn't. It's just kind of, this is sort of what I meant when I said this whole thing is like an interaction of both like information and different chemical structures. So the mRNA contains information because it has a sequence of bases, and that sequence of bases can specify an amino acid, but it's also like a chemical structure. It only allows certain things to bind to it, and based on how that binding happens, we're going to have different proteins that get made. And those different proteins will have different shapes because their primary structure will be different, and they'll all interact differently. And that shape gives it a different function. So it's all about like kind of this interplay of information and chemical structure.
The, on the slide that Buck put around.
Yeah, down here.
Yeah, the
Yeah.
upper right, like the kind of road ribosome,
Yeah.
is this kind of like a sequence of phases, like that's kind of like the termination stage where it's kind of branched off?
Yeah, I would guess either that's one that just hopped off or one that's about to hop on, or
Okay. It's hard, so it's kind of hard, it's hard to say which direction it's going.
yeah, from this slide, yeah, it is. I mean, maybe someone really trained in reading these could tell, like maybe this spacing or the way they're oriented means something, but I don't know that. I don't know if it's known at all. But all right, so just we're gonna run through each of the steps, and then we'll be done. Chapter 12. We're gonna watch our review video and then start our review activity. So initiation. All of the parts of the ribosome are going to come together. So the ribosome is made up of two separate parts, which we're not really going to get into, but they're all going to connect at this initiation. At this at the promoter, or sorry, the start codon. Wow, I'm getting mixed up too. So this is all going to begin at the start codon. That's where this is all going to attach. And at the start codon, we're going to have our first tRNA come in, recognize that start codon, and bring over the appropriate amino acid. So we have 3 sites on our ribosome. We have an A site, a P site, and an E site. And if we can remember what they stand for, I think it's really helpful in knowing what the different functions are. Yeah. So, sites of the ribosome, we have A, E, and E. So these are all, we have these different sections on our ribosome. These are the different sites. So A, stands for amino, P stands for peptide, or we can remember it as protein if you like. Those are really synonymous. And E is exit. So what do you think is happening at the exit site? You think a lot of stuff's coming in that way? No. And the cell follows those rules. So we have exit site where everything's going to be leaving, and that's on one end. So do you think things are going to be coming in through the P or the A site?
A.
Yeah, they're going to come in through the A site. This is where an amino acid will come in for A amino acid. That's where our tRNA comes in with a single amino acid, and then it's going to kind of shift over to the P site, and this is where we're going to get a growing chain of amino acids called like a peptide or a polypeptide chain. So this is all going to be happening at our P site where we're going to have our peptide growing. So after initiation, we have elongation. So it's just going to keep going about its job, bring it over the appropriate tRNA that's going to bind, and we're going to get this growing peptide chain that's going to be coming out from like the P site. So our amino acid's going to come into the A site, we're going to make a bond, and it's all going to shift over one. Or it's going to shift over 3 bases, but one like reading frame. So it goes from like these 3 bases to those 3 bases. It doesn't go like just one base at a time. Like the reading frame is 3 bases at a time, and it just goes to the next 3 bases. Does everyone kind of see the difference between going 1 versus going 3?
So there's only the center do something?
No, it's all 3 bases in the codon are necessary to specify an anticodon and bring over an amino acid.
No, there's 3 pillars,
Okay.
and so it's going to go
On the ribosome?
Yeah.
Yeah. Well, so that's where the center one is where the bond forms between the amino acids. So that's called a peptide bond between this chain that's growing. So that bond is really formed at the P site. But the A site leaves this exposed mRNA to let the tRNA come in. And that tRNA is going to have the right amino acid to add.
And then it moves it to the center pillar.
Yeah. And then at the E site, this empty tRNA is just going to bounce and just go back and grab another amino acid later. All right, and then termination, that's where everything's gonna fall off. So when we hit the stop codon, the ribosome is going to break up, the mRNA is gonna leave, or it's gonna break off the mRNA, and mRNA and the ribosome can go their separate ways, and our string of amino acids is also going to disconnect and fold in its own way and just go do whatever job it was made to do. So that's translation. So that's the last step of the central dogma. So let's do a little bit of review. So what's happening in translation?
Which translation?
What's that?
mRNA is being translated
To make protein. So okay, so in translation, we're going to take our mRNA that we made during transcription, and we're going to translate that into a protein. Or we're going to use tRNA to make that happen. So what did the tRNA do again? So it binds to our mRNA, and it's going to bring over the right amino acid with it as it binds. So that's one type of RNA that's involved in translation. What was the other type that's involved in translation besides tRNA? mRNA. So we have our mRNA that leaves the nucleus, and then our tRNA that's going to bind to the mRNA with the appropriate amino acid. What were the 3 steps of translation? Great, exactly. And so why do we have this triplet code? Why are there 3 bases that we look at rather than just 2 or just 1? Yeah, because we want to make 20 amino acids, and with just 2 bases, we can only make 16 different amino acids, and that's not enough. So we need we need to look at 3 bases and have a triplet code in order to make all 20 amino acids. And so how does translation know where to begin and where to end? What's that? Oh, okay. So those were the different sites in the ribosome. Yeah, and it kind of starts and ends at what's that? Yeah. So it starts at the start codon. That's where the ribosome is going to bind to the mRNA at the start codon, and that's going to be in the P site in the beginning. So we're going to have just one amino acid on a tRNA that's going to be in the P site of the ribosome when it reads the start codon on the mRNA. And where's it going to stop? Stop codon. Yeah, so when it hits the stop codon on the mRNA, it's all just going to disconnect and fall apart.
And if that's the answer to the question, how does it know where to begin? And it just always continues the same process?
No, so where does it start and end?
Does it start where on the end of the stop?
Yeah, so it knows to stop when it hits
How does it know?
when it hits the.
It's always the same thing?
Yeah, it's always starting at the start codon and stopping at the stop codon. Yeah, exactly.
And the
Yeah.
stop codon, is that in the E site?
So that'll be in the A site when everything disconnects.
Okay.
Yeah,
That's when it stops?
yeah. So you'll see in the animation how that happens. So when the stop codon comes to the A site, it's not going to bring an amino acid with it. It's just going to let everything disconnect. When it gets to the shifts over to the P site, then everything's just going to fall apart. All right. Do you want to watch our review video? Also, sorry, when you're studying, you can use this. This is a figure in the book as well. You can use it to kind of figure out what's happening from transcription, how we're processing our mRNA, and how we're going to make proteins from that, how it's all connected. So this would be a good thing to study as well.